is that why?
Yeah, it is. It's too short. Taking attendance takes up too much time. All the other shop talk takes up too much time. When we really get rolling, the class is over. All right, so microscopy. So this is covered mostly in the appendix. So actually, let me go back to that. Right, so Appendix B, to be exact. It was going to close me out because I was inactive. Read, study, practice. Right, all the way down at the bottom of the list because it's at the back of the book, the appendices. We're reading, it's 4 pages, y'all, in the book. Right, online it may seem like more or less. So this, most of what I'm going to talk about, except for the gram stain stuff, which is in Chapter 2, is here in your book. So as they talk about in these appendices, they talk about the importance of contrast and magnification, resolution. They don't mention numerical aperture, and I'm just going to define it for you guys here. In lab, you utilize that term and you do the math related to that. We keep the more complicated stuff to math, like math to lab. So the good news is, lecture, you don't have to do mathematical calculations. And then refractive indices, which they do discuss in this appendices. So we've already looked at that website, right, where we could zoom in and zoom out, right, and see all the way down to a carbon atom for that. On this drawing, you know, compares the relative sizes of several different microbes in a hydrogen atom. So the range of our light microscopes, really, the really large viruses you can kind of sort of see, but for the most part we're just seeing bacteria and larger microscopic organisms like amoeba that are eukaryotic, and even our own cells you can see clearly with a light microscope. If you want to see much smaller microbes like viruses, you've got to go to an electron microscope. And then they have even more fancy that your book doesn't discuss, scanning tunneling microscopes nowadays that can actually see at the atomic level. See individual atoms, yeah. I know. One of these summers I'm going to write a grant and go on sabbatical and go play with one of these. I've worked with most microscopes. I've seen electron microscope used. I've never used one myself. We're hoping to get one here at Delgado. Pretty excited about that. I've worked with just about every type of light microscope there is, though, but I have not the more high-tech ones. So why do we want to use a microscope? Can you see bacteria with your eyes? No. We need it for what? Magnification, right? So these are electron micrographs, right? So these are images produced by specially staining and preparing specimens and using electrons to create an image. So this is a little tiny. So notice the bar here. This is 100, this bar is 100 nanometers in length. This is a bacterial phage. This is a virus that infects bacteria. Looks like a little spaceship, huh? These are kind of my favorite. This is a bacteria, actually probably just getting ready to divide into 2. But you can see these long flagella and fimbriae structures we're going to talk about in the next unit. Under the light microscope, you wouldn't see these much finer appendages to the bacteria. You'd just see the bacteria, and even then they'd look really tiny with a light microscope. This is a special procedure where they've etched away the layers of the bacteria so you can see the different layers. Now sometimes electron micrographs will be colored, just like people used to take old-time black and white photos and they would literally paint or color on them. They do the same thing with electron micrographs. Electron micrographs are grayscale, black and white basically. But then sometimes they'll go in and color it to be able to visually help you understand what you're looking at and point out different characteristics. So it can be kind of confusing sometimes. And that's why, of course, it's always important to read what they write under the pictures as to what you're looking at, right? So the other thing we've got to deal with microscopy is contrast. So they use special stains for electron microscopy? Do you think we need to use special stains for light microscopy? Yeah, pretty much bacteria are translucent. If you didn't stain them, you wouldn't see them. The light would pass right through and you couldn't distinguish them from the water or the glass slide that you stuck them to. So again, we color them. We create contrast by staining them. There are some microbes that have their own pigmentation, but those are rare. For the most part, we're going to stain them, and we're going to even use specialized stains. So most of the stains we use stain the bacteria itself. The stain actually goes inside of them. So you can see their shape. So this is an example of a gram stain, where you actually end up with two contrasting colors if you have the two different types of bacteria. This is an acid-fast stain. Again, you're going to end up with different colors. It's a differential stain if you have the two different types present. Endospore stain, as the name implies, it stains what? Endospores, right? So it helps us visualize endospores. So you see the little red dots are the endospores. The other are bacteria that are still actively metabolizing and haven't formed a spore yet. Sometimes we do relatively simple stains where we just use one dye. So we really can just see the shape and how the cells are arranged. There's another type of dye procedure where we don't actually stain the cells, we stain the background. So this is the bacteria right here, and notice they're clear. The background is what's been stained here. Yeah, this picture. So there's the bacteria. The problem with this one, especially for beginning students, you guys see all these other little specks? That's just crud on the slide, like dirt, dust, pollen. This is the bacteria, these two little guys. And over here, here, those consistent rod shapes. So my students in lab get annoyed with me when I tell them to draw before they came to class this last week, all the things that we're going to look at under the microscope. And I'm like, because I want you to have a frame of reference of what you're looking for. Because some of these slides that we looked at were fecal samples. So in addition to the bacteria, there's literally a whole lot of crud, a whole lot of shit on the slide. And students are like, oh, is this the organism? No, that's literally shit. You got to know what you're looking for, right? And so with simple staining, too, a lot of times, right, you're staining stuff or not staining stuff that isn't what you're looking for. It's good to have a general idea of what you're looking for. Resolution, probably just as important as magnification and contrast. If your image is blurry, is that going to do you any good? No. All right? So I'm going to pick on one of my girls in the back wearing glasses. Did I pick on you for lab? Either one of you. I did pick on you, didn't I? So I know you can't see me, but you've already been through it, so I can't pick on you again. All right. Do you need those to see me? Those glasses? Okay. Can you take them off for a second? Don't tell her. How many fingers am I holding up? Okay. Go ahead, put your glasses back on. She got it right, but I bet you they were blurry, right? She guesstimated, didn't you? Based on the width of my fingers. She didn't actually see that it was 3. Had I gone like this, it would have made it easier for her. Why is that? They're further away. It might have actually made it a little bit harder. Right? Depending. You use more than your eyes to see, right? She figured out, she's like, oh, based on the width, it's definitely more than 1 finger. It looks like it's more than 2. Oh, yeah, I cheated too. She looked at my other fingers, right? So again, you use more than just your eyes, but when you're looking under the microscope, especially if you don't know what you're supposed to be looking at, if it's blurry, this is not going to help you. And this is where what we call resolution comes into play. How clear and crisp that image is. For us with our eyes, you literally have a lens in your eye that you can change the shape of it so that you can see close up and far away. Hers, she can't change it enough to see me up here. So she wears additional lenses to correct for that. As we age, our eyes don't get really good at at doing that, going back and forth, so that you end up wearing things like reading glasses, specifically. You have real trouble at that up close, right? You're said to be farsighted. You can't see near anymore, you can see far. Right? So I call it old woman's disease, right? You're supposed to wear reading glasses. But I live in the technology age, so I just blow everything up on the computer screen. So I have 2 computer screens in my office. Because I run out of room when you blow everything up. This is a problem we're used to nowadays too, though, right? So if you take a picture of somebody, especially with your smartphone, can you blow it up real big? Pretty big, because it stores a lot of information, right? So that you have enough information that you can make it bigger. But if you had one of the old ones, could you blow it up? Not very big. It got what? Blurry. And that was an information storage problem. Where with this, it's a light focusing problem. It's the lenses. And on the microscopes, unlike our lenses in our eyes, you can't change the shape of them. They're set. They're glass. Just like your glasses, you can't change, right? So if your eyes don't change or your eyes change, what do you have to go do? Get a new set of glasses, a different prescription, right? Where they mold that glass just a little bit differently. So for our microscopes, we know the level of resolution because we know what it is set at, what the prescription is, you could say, for our lenses. So this ability to be able to resolve things and them not be blurry is referred as resolving power or limit of resolution. Those of you guys in lab, you can calculate this, right? We know how far apart 2 objects have got to be, what that resolving power is going to be, so that they will look separate instead of, like this image in your book, which I haven't stole yet. See, they're far enough apart. Just like my fingers, though, if they're not far enough apart for the resolution of what you're looking through or using, microscope or your own eyes, then you're going to see that blob, right? Where they're blurring together. Make sense? So with light microscopes, we're limited by using light. Electrons have smaller wavelengths, better resolution, but we can't see electrons, right? We need special devices to create those images for us. So the light microscope, we're stuck within those confines, right? Of how good of a lens we can make and the wavelength of light. That measure of that lens's ability to focus light is called numerical aperture, and it's set for each lens. So each lens has a magnification, and this 0.7 for this one is its numerical aperture. You could say its prescription. So we can, as I said, we can calculate what the resolution is for any one of the lenses and combination of lenses for your microscope. The most powerful light microscopes, about 2,000 times magnified, about 100 nanometers, you can see. All right, so we're going to pick up here next time, but again, as I said, over the weekend, I'm going to be posting all the assignments and the next lecture. I did. Did I screw up? Let me